Chapter 7 will dive into metabolism. We will cover the basics as well as metabolic reactions. So what is metabolism? It's every type of reaction that takes place within the cell. Anabolism specifically refers to those reactions that take small subunits and build larger macromolecules with the use of energy. Catabolism takes large molecules and breaks them down into their small subunits. Energy is released by this breakdown. Some catabolic reactions that release energy are the Krebs cycle, glycolysis, cellular respiration, and fermentation. The products released from these reactions can be used in anabolic reaction to build things like proteins and DNA. You may have heard about enzymes and how they are catalysts. They speed up the rate of a reaction without being used up or becoming part of the products. The majority of enzymes are proteins that may rely on cofactors to function properly. Each enzyme is structurally different and has a specific reaction that it can catalyze. On the surface of the enzyme is an active site where the substrate can bind. They're not used up, rather they are recycled and can catalyze another reaction. The function of enzymes can greatly be altered by both temperature and pH. We discuss regulations of enzymes shortly. In order for an enzyme to catalyze a reaction, the substrate must bind directly to the active site. A conjugated enzyme is one that consists of a protein and a non-protein molecule. They're also called holoenzymes. Here we can see an apoenzyme, or protein, bounds to a metal cofactor. An example of an enzyme like this is the catalase enzyme that we discussed last chapter. Sometimes a protein may have a coenzyme attached as well. Your simple enzymes are solely made of a protein, while the conjugated enzymes that you just saw have non-protein molecules attached. In order for a reaction to proceed, the substrate must come in and bind to the active site on the enzyme. This fit is much like a lock and key. Once the binding occurs, a bond forms between the enzyme and the substrate. The reaction then proceeds and the product is released. Here we can see how the enzyme has a special active site. Not all substrates can bind because it is highly specific. Only our orange-pink substrate has the ability to bind. We see then the products released, and the enzyme is recycled to catalyze more of the same reaction. Last chapter we discussed trace elements that are required by organisms. Here is an example of why they're needed. They serve as cofactors for enzymes and can help to enhance the activity of the enzyme by bringing the substrate and active site closer or by participating in the chemical reaction. Coenzymes are organic substances that can also serve as a cofactor. They can remove and add chemical groups. They're important in cellular respiration as they transport electrons and hydrogen ions. Enzymes are named by their actions, what substrate they bind, as well as the site of action. The prefix usually is determined by the substrate they act on, and the ending is "ase." Let's look at two different types of enzymes. Constitutive enzymes are those that are always present and in constant numbers, no matter how much substrate is available. Enzymes that take part in cellular respiration would be considered constitutive enzymes. You can see that even with the addition of substrates, the numbers stay the same. Regulated enzymes can be turned on or induced and turned off or repressed. If more substrate is added, the enzyme is induced. More of it is made to accommodate the extra substrate. If substrate molecules are removed, then the enzyme is repressed or turned off. As I mentioned earlier, temperature, pH, as well as osmotic pressure can all affect the activity of an enzyme. Alterations make the enzymes unstable. Denaturation can also occur via alterations in the enzyme's environment. Bonds in the apoenzyme are broken, which then prevents the substrate from coming in and binding to the active site. Let's look at the ways that a bacterial cell can influence the activity of an enzyme. In competitive inhibition, the cell can produce a molecule that resembles the substrate. This molecule then binds to the active site, directly competing with the substrate. This prevents the substrate from binding. In non-competitive inhibition, a molecule binds to a regulatory site on the enzyme. It changes the shape of the active site, which then slows down the reaction. In competitive inhibition, we see the active sites, the normal substrate, and the competitor. On the left is what should normally occur. In competitive inhibition, the competitor binds to the active site, preventing the substrate from binding. In non-competitive inhibition, we see the regulatory sites and the active sites. On the left is our normal reaction. On the right, we see the product directly bind to the regulatory site, which changes the shape of the active site. This type of regulation is a type of negative feedback. Bacteria can also control the synthesis of their enzymes. In enzyme repression, synthesis of an enzyme is stopped somewhere in the pathway. The more product given off from a reaction can suppress production of the enzyme. The reason the cell does this is because they already have enough product. During induction, a cell can turn on the synthesis of an enzyme whenever there's plenty of substrate. The normal process is transcription, in which DNA is transcribed into mRNA, and then translation into protein. Enzyme forms and acts on the substrate. The product can then come and bind to DNA and repress the production of the enzyme. E. coli has been well studied. If E. coli is inoculated in lactose, then it will produce lactase to break it down. But if we put E. coli in sucrose, it will switch to the production of sucrase. It has the ability to adapt to its environment and utilize a wide variety of nutrients. In metabolic pathways, we usually see a multi-step series or pathway. Sometimes we see products of one reaction serving as substrates for the next. There are also cyclic forms. Glycolysis is a type of linear pathway where the product of one reaction is a substrate for the next. The Krebs cycle is a cyclic form where the starting molecule is the ending molecule. So where does a cell get its energy? ATP, of course. In exergonic reactions, energy is released, while in endergonic reactions, an input of energy is required. Typically, these reactions are coupled and rely on one another. The energy released is stored between the phosphate groups of ATP. Once the bond is broken, the energy is released. Let's look at two important terms. Oxidation is a loss of an electron from a compound. Reduction is when a compound gains electrons. Whenever we couple the two, we call it a redox reaction. These reactions are very vital to metabolic processes. In these redox reactions, special enzymes called oxidoreductases strip electrons and add them to other substrates. NAD and FAD are important ones in cellular respiration. On your left, you see oxidized NAD that pulls electrons and hydrogen ions from a substrate and becomes reduced. During redox reactions, we often see phosphorylation, where energy is used to add phosphate groups to ADP to generate ATP. When the electrons are captured, hydrogen comes along for the ride. The removal of hydrogen is termed dehydrogenation. ATP, our energy source, and a bacterium's energy source, is made up of a nitrogen base, ribose, and 3 phosphate groups. The energy in an ATP molecule is stored between their phosphate groups. Enzymes are used in both catabolic and anabolic reactions. The electrons and energy from catabolism can be used in anabolic reactions. The 3 major metabolic pathways that we will discuss are aerobic respiration, where oxygen is the final electron acceptor, anaerobic respiration, where something other than oxygen is the final electron acceptor, and fermentation, where organic compounds are electron acceptors. In aerobic respiration, glucose is converted to carbon dioxide and ATP. This involves glycolysis, Krebs cycle, and electron transport chain. Oxygen is the final electron acceptor, and this occurs in most organisms. Anaerobic respiration occurs in those organisms that can undergo metabolism in the presence or absence of oxygen. This too involves glycolysis, Krebs cycle, and electron transport chain, but other substances like nitrates and sulfates are the final electron acceptors. Fermentation does not require oxygen, and organic substances are the final electron acceptors. In glycolysis, glucose is broken down into 2 pyruvate molecules, 2 NADHs, and a net of 4 ATP. However, the cell does not actually get 4 ATP because it takes an input of 2 ATP. So it's like you buy a $2 lottery ticket, win 4, but you actually only take home 2. In the Krebs cycle, pyruvate is converted to acetyl-CoA. In this process, NAD and FAD are reduced and will transfer electrons. 6 NADH molecules are released, along with 2 FADH2 and 2 ATP molecules. In the conversion of pyruvate, 2 carbon dioxides are released. Redox reactions occur, and 4 CO2 molecules are released. 2 ATPs, 6 NADH, and 2 FADH2s. The reason that we have 2 FADH and 6 NADHs is because there are 2 turns of the Krebs cycle. This is because we have 2 pyruvate molecules. In the electron transport chain, which occurs in the cell membrane of bacteria, those NADH molecules and FADH2 act like Ubers and bring electrons here. In the process, hydrogen ions are pumped outside the cell, oxygen pulls at the electrons and hydrogen, and water forms. The major compounds that carry out these processes are NADH dehydrogenase, flavoproteins, coenzyme Q, and cytochromes. Cytochromes assist in transporting electrons from one carrier to the next. Embedded in the cell membrane, we can see the cytochromes. NADH and FADH bring electrons, and they're transported from carrier to carrier. Hydrogen is transported across the cell. A concentration gradient develops, and then ATP synthase pumps hydrogen back in. Oxygen pulls at the electrons and hydrogen, and water is formed. We also have the bulk of our ATP produced during this stage. In eukaryotes, this takes place in the mitochondria. Oxygen is converted to superoxide ions and hydrogen peroxide. We know that in order for a bacterium to survive, they must be able to produce special enzymes to detoxify them. In anaerobic respiration, nitrate can act as the final electron acceptor. In the process, the enzyme nitrate reductase converts the nitrate to nitrite. Some organisms, like Pseudomonas aeruginosa, can further break down nitrite to nitric oxide, nitrous oxide, and nitrogen gas. Bacteria can also break down carbonates and sulfates. This particular reaction does not produce as much ATP as we see in aerobic respiration. Let's look at fermentation. Fermentation can occur with or without oxygen. Organic molecules serve as the final electron acceptor. Only 2 ATP are generated in this reaction. This is useful for organisms that don't have an electron transport chain, or those that do, but are lacking oxygen. Fermentation can lead to the production of alcohols or acids like lactic acid. Organisms can also rely on non-carbohydrate compounds for glycolysis. Lipids can be broken down and enter the Krebs cycle. Proteins can be broken down into amino acids. Their amino groups are removed, and they can then enter the Krebs cycle as intermediates. Organisms have the ability to combine the catabolic and anabolic pathways to enhance their efficiency. The intermediates from glycolysis are used to synthesize amino acids, which then form proteins. Glucose can be used to synthesize more carbohydrates. Amino acids can be deaminated and used in various reactions. The organism benefits from using all reactions together. Let's look at the synthesis of macromolecules. Simple sugars come as nutrients or can be synthesized. They can then be used to synthesize polysaccharides. Glucose and other carbohydrates are important in composition of cell walls, peptidoglycan, nucleic acids, capsules, and glycocalyx. Proteins are important enzymes, and they're part of both the cell wall and cell membrane. Their building blocks are amino acids. Nucleic acids are DNA and RNA and are essential for protein synthesis. Anabolism and catabolism both play roles in the assembly of the cell, whether it be DNA replication, cell wall, or cell membrane synthesis.